This meeting is being recorded.
Okay, go ahead, Mohammed. Thanks.
Okay, thank you, Sergey. Today, actually, we will talk about the multiplexed imaging for brain tissue. I think this domain actually. I think this domain actually is a novel domain that come to the brain tissue, and mainly the need for scanning here. The motivation for that actually is we are actually trying for capture the cell localization and cell type in the brain. So the brain actually have like millions or maybe around 100 billion cells. And these actually together actually drive our ability for thinking, remembering, and feel emotions, which actually these cells is actually consist or make us who we are. So the need actually to try actually to see or investigate these cells in the brain is huge. Our main cell in the brain is the neuron, and there is actually multiple other types that actually beside the neuron, like the astrocytes or the other type, these actually shape or contribute a critical role in maintaining our proper neural function. So the multiplexing technology actually come here to give a hand of that. So it enable, in general, the biologists and specifically the neuroscientists to measure multiple molecular targets in the same time, simultaneously in the same or in a single experiment. So these techniques actually profiling individual cell in the context of the tissue and providing a snapshot of cellular function. Many of the techniques actually that is using the multiplex technology is antibody-based detection of proteins. So we have like a panel here of proteins and antibody that can be associated with each cell to determine the cell type. For example, here we have the neon, which is a marker that actually can actually localizing the nucleus of the cells. So these actually, there is like multiple technology have been recently developed that's ranging from using the protein panels to the RNA panels and give us an imaging in most cases. So our rules here can actually come to analyze this image and try to find like the biomarkers that can identify a certain function or a certain disease. Here an example actually of two of these technologies. Codex is a recent one that come, I think, from Stanford University, and it uses a protein panel mainly and have the ability actually to get maybe like 40 different protein in the same image. So the idea, if we get the protein here, we can go to the spatial proteomics, which actually an imaging based spatial proteomic method enable quantitative and spatial analysis of over 40 protein markers across a whole tissue section at single cell resolution. And this spatial proteomic is a large scale analysis of proteins and their localization and dynamics within tissue. Maybe we can start from one single omics like proteomics here, or we can actually integrate it with another different omics domain like genomics to get more comprehensive insight into tissue biology and discover a novel biomarker signatures. So the pipeline here, in general, we can see that there is in the beginning an experimental design, and then based on your need from your experiment, there will be a staining and imaging stage using some technology, some microscopy technology here, and some antibody and antigen panels based on your requirement. And this is actually will end, this step 1 and 2 will end you that you have an imaging in most cases, like in the codex cases. And from there, actually, we can start our data analysis and insight generation. So our case of study today will be a paper published in Nature Communications, which is World Brain Tissue Mapping Toolkit using large-scale, highly multiplexed immunofluorescence imaging and deep neural networks. So this actually paper came last year, 2021. And the authors in this paper trying to address mainly the challenging to bring the multiplexed imaging to the brain tissue. There is actually different stage this paper try actually to describe. One of them, when they are trying to overcome the problem that the brain tissue is actually considered as a large scale comparing to the other tissue of the body. So there is a need that we need when we get a slice from the brain, we need actually the whole context analysis in order to get the right insight of the cell in this on this tissue, I mean. So this actually required from the authors to validate or using a validated immunolabeling protocols that are capable of imaging large panels of biomarkers across this large brain tissue. There was also a need that based on that first step, there is a need for multiple channel corrections and registration to eliminate all the noisy structure that normally will actually produced on the imaging output. And the author actually provide here a sample, a protocol for sample preparation, and they actually show all the pipeline till they get actually their tissue ready for analysis. So an overview of the whole brain tissue phenotyping pipeline that used in this paper is showing here. I will talk now about the upper part, which actually consists of a raw high content 2-dimensional multispectral imaging data set. So they used actually that. And from there, they start to make a batch of slide scanning at 10 micromillimeter or 10 micron from an animal actually brain. It's a rat brain actually. And these sections actually get feeded and started to be registered and corrected. And in the last, they actually work in trying to localize the cells in accurate mode. And then from there, they actually try to classify these cells. So as a first step, they actually used here a 10-plex panel for 10 different markers that can be actually localized in the rat brain. We can see here in the top, we can see the microscope, the optical microscope they used. Its size actually. So based on that, they have different wavelengths and used in this microscopy in order to capture different cell types based on the antigens of these cells and the antibodies used in this experiment. So coloring or actually giving different differentiation for each cell actually is a process that can be done automatically during the experiment and can be done actually during the image processing in a later step. We can see here different markers. For example, the first one on the left, DABI marker, which is actually a marker for the nucleus of the cell. And there is actually emerging image on the right showing what could be the output or the final output of this microscopy. And actually, this is not come in a single channel. I mean, no, actually, every channel represents a marker, and there is a stack you will have in the last. That this stack actually have multiple channels, like here 10 channel, for example, and the image processing actually can extract each channel alone or mixing different channel or fusing this channel in order to give you more insight on the data. So the part that after this image acquisition is dealing with mainly with the correction. So the post-acquisition image optimization module here is given, and they start in the beginning doing a registration for the different channels. And they actually use here a middle level image processing technique, which is or the oriented fast and rotate brief. Its abbreviation is ORP. It's similar. This one actually is similar to SIFT and SIRF, and more easier actually, and have multiple implementation. So on the left, we can see the here the key points actually is identified between the two channels, and the registration actually shown after and before the registration part and after the registration parts. Next, actually, it comes to the part that we deal with the intra-channel correction. So we have here like multiple type of noise or distortion. For example, the uneven illumination, which actually show us that the light actually doesn't illuminate on the parts of the tissue in this case. There is also multiple source of noise, like the autofluorescence, that is a fluorescence come naturally from the substance without even attach any antibody for this substance. And there is also the tissue folding, and there is also the photo bleaching, which is a type of fading happen in the fluorescence signal. So actually these techniques, this noise or this actually distortion needs some pre-processing here in this stage. They mainly used a technique called alternating sequential filters, and it give here actually much better results in the bottom, in the middle. And for the inter-channel correction, which is actually a problem can happen from mixing the signals of different channel, because we are dealing here with a very thick layers. So in this case, actually, we can see a noise come from the neighbor channel affects the original signal that we have in the current channel. So they use actually here a semi-supervised sparse. I'm sorry? Yeah, so they are used actually here as semi-supervised sparse linear spectral and mixing approach, which actually, I guess it's like a type of independent component analysis to actually separate the signals of the two sources. And we can see also, again, the results in the bottom, which more accurate in this step. So based on that, they actually moved to find the bounding box that can identify the cells in this tissue. One of the things that actually hard in this domain, that there is a huge overlap between the cells. The cells are very crowded, and it actually needs some specific techniques in order find a true or proper isolation between the cells that are very close to each other. So they actually, in this case, they used the faster RCNN as a real-time here object detection methods that can propose multiple region, and based on that region, actually, they actually can localize the cells separately. So they actually developed, in this case, a transfer learning based approach to generate a sufficient training sample to train the faster RCNN, and was able actually to detect the cell based on that technique. The network generates a set of bounding boxes indicating the detected cell nuclei, as we can see in the middle of this picture. However, they found that the accuracy here was not actually high when they depend only on DABI marker. DABI for the nucleus is a nucleus marker. So they actually found this is not high accurate. So they decided to combine that with another complement marker, which actually in this case we can see it's the pan-histones marker. So the results actually in this case, when they are combining both, as we can see from the graph, it actually gets much better accuracy. They achieved when they are using DABI alone, it was like 0.88 area under the curve, and when they are using the pan-histone alone, it was 0.8 alone. But when they combine both, actually it gets like 0.97. So this actually step, they use it in the next part when they're trying to make classifications for the cell. So we can see here they have they have actually two approaches here to use. One of them is a threshold based. So they go to the intensity of the cells and using this intensity as an indication for the cell type. So for example, if there is a certain cell have a high intensity in a certain marker, then they can use that as indication of the cell type. But this actually approach, according to their study, wasn't the best approach to have. So they tried to use a type of network here, which is the capsule network. Specifically the improved version of the capsule network, which actually is mainly using, it's like the neural network, but instead we have a neuron in the hidden layer that can take a scale or a multiplication of the weight just as a scalar in this point. No, they actually use that neuron as a part that can actually get a vector of feature, not just a scalar. So this encapsulation actually, or capsulation actually, using in each stage, in each neuron, a vector of feature, a complete vector of feature. And the result actually, it come out in more accurate classification model. So as we can see from the last row, this is actually showing the difference between the thresholds and the capsule network. So the graph here for the threshold part actually showing you there is an overlap in the intensity. There is a signal here actually should be indicated as or showing that this cell that have this signal shouldn't be actually classifying like a astrocyte, for example. But because there is an overlap here between what can be positive, true positive or no, this is actually make this overlapping affect the final results. But in the other side, we can see that the true positive and the true negative for the same cell type actually have a there is a margin that actually can separate between them based on this capsule network. So this is actually result in more accurate results in identifying the different cell types or the different mean cell types in the brain in this case. And we can see here like 4 or 5 mean cell type that they are working on classifying.
Hey, Sergey.
Yes.
Sergey, is he still there?
Yeah, I'm here.
Yeah, so basically this is like one of the main reasons I was interested in this paper when I was talking earlier is because it's like the first paper I've seen where the capsule network's been applied.
Yeah, exactly. That's my thought is like, did they get capsule networks to actually work on something?
Yeah. So that's kind of like last time we talked about capsule networks, everybody wanted to know if there was anybody that had made it work. Sounds like somebody has.
Well, the question here is, did they really need to use capsule networks because nothing else worked? Or, you know, it's kind of complexity biased. Let's use a complex model and see if it works, and it kind of worked for them. So that's interesting.
Yeah, so actually the main reason that they don't go to any other classification model because capsule network have a feature that it is actually reliable to the orientation of the inputs. And meanwhile, actually, it also have its ability for the open set. So there is actually can be a known type produced from this cell as they are indicated. Beside the mean types, there is also unknown types if there is no high probability for any type of the other cells. So actually, they actually work here on that part in generating the training set that is used with this capsule network from the cells that already have a high expression of their protein or high expression of their markers. So this part come from the thresholding part. So the thresholding part actually, as we said, there is an overlapping between the true positive and true negative. But for the high scored true positive, they use this part actually for training this network in the beginning. And the final part actually they covered in the paper after they get now the bounding boxes of all the cells or the detected cells and get the main types on the tissue. They actually go to get the subcellular compartment of each cell, which include the nucleus and the soma, the membranes. And they make a quantification for all these type and subtypes, as we can see on the table on the upper right hand. We can see this is actually showing the different cells that they had in this actually this section. And in the bottom we can see actually in the middle, sorry, we can see actually 4 different subtypes from the main types that they actually said they could actually in a reliable way they could extract according to their model. And in the bottom we can see here a heat map can show us, for example, in the middle, we can see a heat map for the neuron. And this heat map actually showing the density of the neuron according to the a megapixel area. And in the last one, we also see another type, oligodendrocyte for the same heat map and the same density for this megapixel area. So in general, the author in this paper put like a protocol that can be working if we want to bring the multiplexing imaging technology to the brain. And this actually consists as a part in the beginning that we need to partition the brain, and then after we get the imaging complete, we need again to reconstruct and complete the image mosaic in this case, and based on the layers that we can construct, we can also complete our data set for analysis. So this is actually what all we have. They gave also some table showing here, for example, the different biomarker or different marker that they have in used, and their actually association with different cell type. And we can go particularly to table number 7, which actually showing how they can actually report the final results according to their protocol. So each cell actually have a location based on its X and Y in the tissue, and there is actually also an indication of the biomarker it has. So based on the unique biomarker it has, or based on the combination of biomarker it has, we can actually classify the cell. And based on this actually coordinate and this cell type, we can actually know which cell has which neighbors. And from there, actually, we can make more analysis on that part. So this is what they have till that point. And if there's any question now.
Yeah, thank you, Mohammed. Everyone, questions or comments or discussions? So I have a technical kind of question. So you need to cut the slices of the brain, right? And then after you cut the slices of the brain, the way they show, or I don't know, are they trying to put the slices back into the 3D structure, or it's only looking at one slice at a time? Are they creating the 3D view of this whole structure of the brain?
Well, actually, they make a 3D analysis, as we can see. It's not a structure. I didn't see a structure actually they have, but they have actually each slice from the brain, and they try to plot on a graph here on the second row on the right hand. They try to plot actually for each slice the cell counts. So I didn't see actually an evidence that they tried to in another way to make, because there is actually a 3D multiplexing imaging. This is a little bit different technology or advanced technology. They didn't actually use it that, but they trying to actually to tell how we can use all the stack of slices to get actually a more insight data that contain all the brain cells, as we can see here. This is all that they did with this 3D actually.
So the goal is not 3D. The goal, they can meet their goal by just looking at the slices, right?
Yeah, they take it slice by slice and plot each slide, what is the count of each cell, and put, I think they take the average at last, something like that, I mean. Yeah.
How large are these data sets typically? It seems very expensive to generate.
How large? So actually, it can be, yeah, it can be actually large because each slice we need to find all the markers in this slice. So we can, for example, in this paper, we have 10 plex, 10 different markers have been used. So for each slice, we need different channel to represent all the cells in this slice based on our experimental design. So yeah, I guess it's if we talk about for example, in average can be, we have like 100 from 100 to 200 slices in the MRI. So in this case, actually, we talk about multiply by 10. And there is also something here different than the MRI we know. This is a very high resolution. It can be like maybe in average like 10,000 pixel width by 10,000 pixel height, for example. So the slice, only the one slice here actually can be like up to 1 gigabyte. Because I see a stack of 40 channel can be up to like 30 gigabytes. Yeah.
And can you use the results in your Histo.js display? Like, is it stuff that you can display in your browser application?
Yeah, this is actually possible if this data is available. I think this data is available somewhere, but we need to tolerate a little bit Histo.js in this case because we have like a more depth slides here. We have a more depth. We have maybe not only 40 slices as we have in Histo.js. This is much more deeper, but it is in the same pipeline, can be integrated. Yeah. The same idea. Yeah.
Well, thank you. Any other questions? Okay, if there is no questions, we can finish early this time. And next time, Farfalla, you're presenting, right? Let me stop the recording. Bye, online viewers.